Good day. Welcome to the BSIG Webinar Series, Episode 5. I am your host, Seth Cordes of Uncommon Catalyst. I'm joined today by Rafat Zaini, research scientist at Worcester Polytechnic Institute. Our guest today is Ken Cooper. Ken received his bachelor's from Massachusetts Institute of Technology and his master's from Boston University. With a career in systems dynamics spanning over 4 decades, Ken Cooper has led hundreds of modeling efforts. While at Pew Roberts Associates, Ken pioneered the development of project management modeling, dispute resolution with systems dynamics. He also led corporate strategy work for senior clients in several industries, including aerospace, automobiles, banking, bioscience, construction, shipbuilding, and telecommunications. His clients have included MasterCard, Fluor Corporation, Raytheon, Boeing, Northrop Grumman, Ford Motor Company, IBM, and many others. Ken is a 2-time Edelman laureate for his modeling work. In 2010, he won the Systems Dynamics Society Application Award for his modeling efforts with the Fluor Corporation. In 2011, Ken turned his focus to human biology, following a lifelong dream to apply systems dynamics modeling to understanding and curing the most complex diseases of the human body. This work led to the first clinical trial ever approved in the US based on computer simulations. Ken has led the development of models of the human immune system, HIV, type 1 diabetes, cancer, and chronic traumatic enthocapopathy. Ken, please correct me on that one. I didn't practice the pronunciation. Today, we'll look at Ken's modeling work on the human immune system and discuss the novel success that he and his team have had with this approach. We've set up a chat window on YouTube. So if you're watching via that, which everybody is, please post your questions there. We'll have some time at the end to go through them on the air. With that, I will introduce Ken, or hand it over to Ken. And all right, Ken, I'm going to switch over to your video now here.
Thank you, Seth, and good day to everyone. It's a pleasure to be with you today. Let's get this working. Seth, I am having some difficulty getting a screen share.
There's your video.
Okay, thank you. Try this again.
All right, now if you switch over to your PowerPoint, you should be able to bring that up.
All right.
There we go.
Very good.
Screen share.
Excellent. So today we're going to discuss what I believe is a momentous opportunity for the convergence of system dynamics modeling and human disease research. Here's the situation that we have today. Every year, tens of billions of dollars are spent on pharmaceutical research. And it's, I think, universally recognized that the whole process is too costly, with somewhere north of $2 billion invested for every successful drug that makes it to the market. It's too long. It takes over a decade to get new molecules to the market. And it's too ineffective, with a very low success rate. And despite what impression might be left by the seemingly omnipresent drug commercials, there are only about 20 drugs that are approved each year by the FDA, and that's for all ailments, disorders, diseases, all of the cancers and autoimmune disorders, all the viruses, cardiovascular issues, neurodegenerative disorders. For all of those diseases, somewhere near 20 new drugs a year. So clearly the challenge is large. But what if you could simulate those human disease conditions and do so in a very short time, likely in a matter of minutes, for decades-long chronic conditions? What if you could test leverage points identify in advance what the most promising targets for pharmaceutical treatments are. And very importantly, as we'll see as we move through the next hour, being able to test combinations of treatments, to be able to test them over the long term, to be able to test them in even extreme dosage conditions without safety concerns, and identify in advance potentially useful combinations of treatments. By doing this, you could rapidly screen thousands of options to increase the value of the research targets, and I believe significantly reduce pharmaceutical development time and cost, and improve the probability of success. In doing that, I think there's an opportunity for the system dynamics modeling community, and perhaps the modeling community in general, to help prevent or find cures for the most devastating human diseases. The amount of societal and economic benefit from this would be enormous. Today, I'll go through a brief sampling of some prior modeling that I've been involved in on the immune system, HIV in particular, type 1 diabetes, cancers, and chronic traumatic encephalopathy, which will probably shorten to CTE from this point forward for ease and speed. And we'll spend some time, I hope, at the end talking with everyone about the potential for this, the huge potential going forward. In going through all this, it's not my purpose to teach. Many of you watching and listening will know far more biology than I am. A modeler, not a medical scientist. But the more I learn, the more I'm convinced that this is an exciting and important opportunity. So, starting where my work started in this arena, why HIV/AIDS first? Well, the HIV research of the last few decades has provided a lot of the scientific knowledge about our immune system, and it was with the immune system that I wanted to start the modeling work in the human body, for a number of reasons. The HIV work and research provided a lot of data that we could use for developing and validating the model. We had the good fortune to have to be able to work with some top HIV immunologists, and we thought even in the early going, and I think it is proven true. And importantly, HIV is far from yesterday's news. There is no cure. 35 million people live with HIV now, and it accounts for nearly 5,000 deaths every single day. So why has it taken so long to get to a cure? There have been hundreds of billions of dollars spent on HIV research. Well, HIV affects either directly or indirectly, it interacts with almost every part of the human immune system. Now, in that beautifully elegant human immune system, this elegant control system, despite the high degree of feedback in that system, in the first talk that we had with the CDC immunologists with whom we worked, we asked, well, how does science currently address the operation of the system and its interaction with HIV. And she said, well, not very well. Despite the understanding that it operates as a system, the scientific community really works in what she called silos. And what she explained to us was that, well, with the many elements in the immune system, the scientists who work on with macrophages don't really speak with those who are dealing with the innate immune system's natural killer cells, and they don't speak with the B cell people, and they don't really coordinate with the dendritic cell people or the T cell people or the CD8 T cells scientists, and on and on. Despite the fact that the virus interacts with so much of the immune system. So to make this point, once we put together an initial mapping of the immune system, which is not intended for you to read, it's simply there for you to absorb as a lot of interacting elements. We use this graphic to pose the question, do we really think that there is a single magic bullet out there that's likely to be the one special intervention that's going to correct the behavior of this entire system that is wound up in the impacts of HIV? I had a map on my wall, a chart on my wall that was half the size of the wall, with a rendition of this causal diagram on it, and I circled in red here the focus of where all of the current treatments are focused. The standard antiretroviral treatments are all focused on inhibiting the entry and replication of the virus itself over that little area where the circle is, and very little goes into looking at all the rest of the elements of the immune system. But what simulation ought to be able to provide, and I think can, is the ability to look at a number of different possible interventions and combinations of interventions that may well be useful in correcting the behavior of the system and improving the immune system, so that the system itself, much as we're seeing with cancers, and we'll talk about that in a few minutes, can operate more effectively to control or eliminate the virus. Now, just to put all this in context, the very first diagram that I put together on a 5-hour flight to Atlanta is pictured here. This is the one that I set on a sheet of notebook paper in front of the person who came to be our principal immunologist. And while it wasn't exactly right in depicting all of the stocks and flows involved in the HIV process, it was enough to convince her that we were onto something as an approach to understanding better the dynamics of HIV. What that became was a much more a larger and much more complex model that simulates the behavior of many immune cell activities around the body, in different parts of the body, through the bloodstream, in the infected tissues, dealing with the host of different actors in the immune system, from macrophages to dendritic cells and NK cells, CD4s and CD8s, B cells, antibodies, and the virus itself, of course. In each one of those segments, there is a cause-effect structure that simulates for each type of cell and the interactions among them, the activation and behavior of each of those cells, from the macrophage and NK cell components of the innate immune system to the adaptive elements of the immune system with B cells and T cells, antibodies and the like. The thing that we chose to do early on was to ensure that we could use a lot of that data that was available about HIV to build confidence in the behavior, in the accuracy of the model. And so we had there's a lot of data from different sources on over the course of time on the viral load in the bloodstream, and the simulations here shown in the blue charts, the CD4 T count in the blood in the short term and in the long term. And we also used the model in building confidence in its validity to replicate a large number of clinical trials that had been conducted over the prior years. And as we were able to do this fairly accurately, replicating all these different trials of different treatments with the same model, obviously, that was one of the key things one of the key steps that gave us some confidence to start searching for other possible remedies for the virus. And so we conducted literally thousands of what-if simulation experiments. And in the course of doing, and many of them automated, but in the course of doing that, those many combination tests, we actually identified many combinations of so-called levers that in the simulation would drive the viral load to or very near zero permanently. So there was no difficulty finding a host of theoretical solutions to accomplish that. In the next few charts, again, against this vague map in the background of the immune system elements, just to give you a sense of a few of the kinds of combinations that were found from the simulation experiments. The size of the green circles that are shown reflect the sort of the strength, or if you will, the dosage of that factor in one successful combination, successful in terms of driving the viral load, the HIV viral load to zero. And so there, one combination there, a larger number of interventions, but with smaller dosage. Another combination that included some work with dendritics, some impact on dendritic cells, and only to convey that there are several combinations of levers that we could find that would theoretically, in silico, if you will, reduce HIV viral load to zero and keep it there. In other words, a functional cure for HIV. But that doesn't mean that there are compounds or chemicals out there that will accomplish those different the levers that need to be pulled. We chose of all of the different combinations that we identified, we chose one for which we found already FDA approved drugs that seemed to pull the right levers, the ones that were identified in the simulation as a rapid yielding a rapid reduction of viral load to zero. That combination became the protocol for a clinical trial that we conducted with a very respected group of investigators. Conducted a multi-month trial. This was, as Seth mentioned, the first clinical trial, to everyone's knowledge, that was ever approved based on simulation modeling results. It was the first HIV clinical trial in which patients were taken off the standard antiretroviral treatments and placed exclusively on our proposed combination treatment or placebo. Such was the hope, the appeal to the scientists involved, that they did so. The absence of news from that will already tell you that that did not succeed in finding a cure for HIV. What we found in the blood tests that were subsequently performed is that one of the key ingredients, one of those green circles, it was the drug that was intended to induce HIV out of latency from cells, a drug called vorinostat, failed to do what we hoped it would do. This was despite what the leading scientists in the world expected from it, but it did not yield the hit on the lever that we wanted. And so without that lever pulled, the trial failed. We predicted that it would fail if it did not succeed in doing that. But the good midterm news in this is that that very combination approach that we identified in the simulations is now being pursued in other HIV trials. So did we find a cure for HIV? No. Did we show that? I think we did, however, show that simulation can be an extremely effective and rapid means of getting to an identified combination that's worthy of testing in humans. To switch course, I want to cover a couple more applications of system dynamics to human disease, only as a means of conveying the breadth of possibilities. And I think this only touches the surface of the possibilities. We built a pilot model, not one quite so elaborate as the HIV model, but of one autoimmune disorder, type 1 diabetes. And in it, we drew upon a lot of different writings. I like this particular graphic from Van Bell that describes the evolution of type 1 diabetes over the course of many years. In this case, it's the relapsing remitting form of type 1 diabetes, and talking about the destruction of beta cells, the loss of insulin production capability, and the development, obviously through the development of autoimmunity, and the very simple summary of the model that we built, pictured here, dealing with the development of autoreactive effector T cells, the destruction of beta cell mass and reduction of glucose production, the eventual development of hyperglycemia. We use that model to simulate the multi-year progression from the first inflammatory response, and then over the course of many years, the slow degradation of the beta cell mass, and in this case, actually, the relapsing remitting form, where the beta cell mass drops, has a slight recovery and plateau, drops again, once again, and eventually gets down to a point where insulin production is no longer capable of controlling blood glucose. But take note that in the very latter stage, there's this little plateau here, and this is a phenomenon that's observed in type 1 diabetes patients. It's referred to as the honeymoon period, where there is a brief stage of hyperglycemia, and then it goes away, or seems to, and that's just that slight recovery of the beta cell mass and insulin production near the final stages before it drops off to permanent hyperglycemia. That's also shown here in this this chart that's from the simulation. Also, in this case, the plasma glucose levels, where the initial inflammation occurs, and then about a decade later, a spike in plasma glucose, the beginning of hyperglycemia, and then this reduction here equates to that same time frame where the beta cell mass has a modest recovery before the beta cell mass drops and the glucose levels reach a higher sustained equilibrium for the duration. Now, what's driving those that slightly oscillating downward behavior is in part a battle in the immune system in between autoreactive effector T cells and Tregs, regulatory T cells. The regulatory, the effector T cells in red here, and the regulatory T cells in blue, where the effector T cells are actually attacking the pancreas, and the regulatory T cells are doing their normal job that they are intended to do, which is controlling against an overreaction of the effector T cells, and so that slows the the destruction of the beta cell mass by the effector cells. Then the regulatory T cells ease up, the effector T cells surge back upward, and so on. That cycle continues for many years until you get to the point where the beta cell mass destruction is sufficiently bad that there is that you go into hyperglycemia, but that shifting balance helps explain, and in this relatively simple model, drives that shifting balance in the relapsing remitting course of the disease. Now, we obviously would want to conduct a number of experiments with such a model, and did so. And in the course of that, the beyond the simple base model that spans about 20 years here, we found several conditions in factors tested that would yield a much later onset of the disease, and some conditions that prevent the development of hyperglycemia or in type 1 diabetes in this benign case. Clearly, these are the kinds of what if simulation experiments that should lead to identifying possibilities for treatment. Perhaps some of those characteristics might be characteristics of individuals that leave them more or less susceptible to the development of type 1 diabetes as a result of somewhat different behaviors in the response of the immune system. And that, I think, is indicative of the great hope that system dynamics and modeling provide for examining and finding potential solutions far more rapidly and effectively for human diseases. I have a couple more examples. Another theme that we've seen in this is that, perhaps looking at the dynamics of disease progression in this way, there may be dynamics that really are quite similar among autoimmune disorders, for example. Does the relapsing remitting form of type 1 diabetes bear some similarities to relapsing remitting multiple sclerosis? Without building the model yet, I don't know, but I'm willing to bet that there are some very important similarities in the dynamics, in the shifting balance and the behavior of the immune system in that form of autoimmune disorder, whether it's type 1 diabetes or multiple sclerosis. Are there dynamic insights into the cause and treatment that may be applicable across multiple disorders? Similarly, in some early look at cancers, we developed a pilot model, if you will, of tumor development and metastasis, and in which we simulate the growth of a primary tumor and the genetic instability that enables that, the angiogenesis that supports the growth of the tumor, and the innate and adaptive immune response. Now, the thing about the immune response to a tumor is that not only does the control mechanism of the immune response fail to stop, when it fails to stop it, it's not only failing to stop it, but some of the responding immune cells actually can join in the tumor, growing it. And in addition to growing its size by virtue of containing immune cells as well, it also actually destabilizes the integrity, the structural integrity of the tumor. And what does that mean? Well, it means that the response of our immune system can actually promote metastasis, allowing parts of the tumor to break off and move elsewhere in the body. Now, that very same set of mechanisms means that, well, if the immune system is misbehaving in some ways, so to speak, in the response and the effect on the tumor and its metastasis, then immunotherapy interventions could theoretically can change that balance with a more aggressive, or really what that means is less inhibited initial response, and a less destabilizing response in metastasis. And it's that less inhibited initial response that is the subject of, excuse me, a lot of new therapies that are in the works now and yielding results in clinical trials. Immunotherapy, immuno-oncology is and has been for a few years the single most exciting and promising area of cancer research. Once again, with the pilot simulation model, just as a demonstration of the kind of potential that such a model could have, we varied a number of different factors and conditions in the model in a series of sensitivity tests and identified several different modes of behavior from this this model of tumors. In one case, the case is labeled as A, running along the bottom of the chart here, this chart spanning many, about 20 years. There is virtually no growth of the tumor. In the extreme opposite, those labeled with a B here, there is extremely rapid growth of the tumor, and you might that might, for example, be typical of what are usually very rapid, rapidly growing tumors, such as with pancreatic cancer. In some cases, somewhat delayed growth, and those labeled in the area with the C, growth to a controlled level, and in some cases, oscillating levels of growth. The point being that that with a system simulation, I think we can. It seems like it would be possible, likely, that we can identify what immune and tumor conditions would cause those different modes of behavior, and in turn, that should advance our progress toward cures. A couple of initial observations from this very preliminary work in modeling cancers. It seems as though the dynamics of the development of tumors and metastatic processes are likely similar across what are often viewed as many different cancers. Different in the sense that they're traditionally thought of as being peculiar to a location in the body, whether that be the pancreas or the kidneys or the lungs, wherever. So much work goes into those individual, in this case, silos for different locations of cancers. But it does imply, or it implied to us when we were doing these analyses, that very similar immunotherapies could be effective across what are traditionally viewed as different cancers. Now, this actually is just what's come about in recent years. Years after we did those simulations, that kind of broad-scale benefit from immunotherapies began being observed in multiple clinical trials. And the latest, I couldn't help smiling earlier this week as I was polishing off some of the material here. One of the early checkpoint inhibitors that's been used in immunotherapy, Yervoy or ipilimumab, that was first developed targeted at melanoma, is, along with other checkpoint inhibitors, is now, as of a few days ago, announced as being somewhat effective in controlling lung cancer and improving survival rates for lung cancer. I think an important piece of proof that immunotherapies can be broadly effective if only we can find just the right targets in the system to hit with those therapies. In these cases, most of these immunotherapy drugs are alleviating the inhibition of the immune system, those regulatory functions in the immune system that control how strongly the immune system responds. Those regulatory conditions are being alleviated somewhat in a classic control system behavior. The final example here I'll offer is from recent National Football League U.S. football fame. In the last few years, there's been much talk of chronic traumatic encephalopathy, or CTE, and all of that talk has focused around concussions. And again, a few years ago, when we were looking into this in conjunction with the immune system modeling work, we came to learn that, as many of you will know far better than I, that the immune system activity in the brain is somewhat different than in other parts of the body, but there are also some basic control mechanisms that are pretty similar. And we wondered if, as with cancers and autoimmune disorders, that the misbehavior of the immune system might contribute to CTE and perhaps even other neurodegenerative disorders. So we put together a pilot model of CTE, and it simulates what's termed as the transdifferentiation of macrophages, an immune cell, into microglia, which are immune cells in the brain, and their function is to clean up damaged neurons. So when there's a trauma to the head, a traumatic brain injury, large or small, these microglia get activated, and they produce cytotoxins that assist in the cleanup of damaged neurons. And the microglia enter a repair stage of their function before, after they do their job, returning to a resting state. So you can imagine the stock and flow structure there of microglia moving around those different stages as they get activated by the need to clean up damaged neurons. And if there are more damaged neurons, well, they induce the resting microglia to become activated, produce more cytotoxins, clear the damaged neurons, and so on. And so if you have repeated, even mild head trauma, it puts that cycle into either frequent or perhaps even continuous operation. So these are simulation results that we're going to look at for a couple of minutes. How could it be that the immune system response to even mild traumatic brain injuries or moderate traumatic brain injuries contribute to ongoing brain damage long after the incidents have stopped, which is, of course, what is observed in the development of CTE. Years after, in this case, football players' injuries have occurred on the field, many years later, the signs of CTE show up in the form of dementia and depression and behavior disorders, and only after, only post-mortem can at this point, can CTE be visibly seen in the brain. So what we have here on the left is a series of weekly head trauma events that we've specified in the model through over the course of about a half a year, and then expanding the time frame to about 10 years. You can see that in some cases there is the development of, this is a chart of neurons destroyed, a growing number of damaged neurons years and years after the head trauma. In some cases, that damage is less. In some cases, it's very small and much later. But again, we can identify which factors contribute to those different time frames and degrees of damage. So we can also see that if there are just a few TBI events, the neuron damage is limited. This is the input of a few TBIs. This is the resulting neuron damage. This is the lower left is the amount of repair that's going on of that neural damage. And in this instance, with the neural damage repaired after a few TBI incidents, there's really no significant long-term damage or CTE. But with a lot more hits, as we saw in the earlier chart, the neural damage stays at a fairly steady rate, and then ultimately begins this explosive climb after a while, and it's at that point that it exceeds the ability of the microglia to repair the damaged neurons, and that in turn triggers a cycle of long-term damage that becomes observable as in its behavioral and cognitive impacts many years later. Now, 3 years after we did these simulations, just a couple of months, I think about 1 or 2 months ago, the preeminent research center in the world on CTE at Boston University announced findings that showed that an accumulation of mild TBIs, not just concussions, cause CTE. And this was a big announcement and very important, one that was not surprising based on the modeling, but again, I think indicative of the kind of insights that we can gain by taking the approach of simulating such chronic diseases. What we found in the simulations as to why that those mild TBIs could accumulate to or could trigger CTE many years later is that very slight imbalances in the control response of the microglia can cause some collateral damage to neurons. And when that happens, it triggers more activation of the microglia, more collateral damage, and snowballs. And that little imbalance triggers this continuous operation of a damage, repair, damage cycle that accumulates over the point where cognitive difficulties become obvious many years later and physical damage to the brain is visible post-mortem. A final footnote to all of this. Interestingly, I thought that the same misbehaving microglia that we just spoke about were identified 2 years later last year in a major medical breakthrough announced by in MIT Technology Review by some Harvard researchers, what they termed microglia as what they called rogue immune cells that were likely responsible for the progression of Alzheimer's and probably other neurodegenerative disorders, just as we identified for CTE. So just as with cancers and autoimmune disorders, there seems to be a possible commonality in the immune system dynamics of CTE that may be shared with other neurodegenerative disorders. Winding up here, I pose the question. I think the answer is yes, but is system dynamics a good methodology to add to human disease research? It seems so because of the prevalence of feedback-intensive systems, the need for a systems view, not just silos of research, the need to analyze dynamic behavior over long time frames and do so rapidly, the ability to test sensitivities and intervention options, very importantly, the ability to test combinations of treatments and interventions, and foresee unintended consequences from those treatments, and indeed to examine alternative hypotheses about how the system works, because we don't know a lot of things about human systems, and the ability to test in silico how alternate theories, hypotheses about how the systems work should be important. There's a theme of commonality of dynamics that emerges time and again in this early modeling of chronic disorders, suggesting important systemic similarities in disease dynamics, and thus possibly in the nature of effective interventions in the form of treatments and cures. Is it possible? We think yes. We think the whole immuno-oncology world thinks yes, that that human system dynamics modeling could inform us as to how we could help guide the body's own immune control mechanisms to help halt or cure diseases. We think the key message is the immense benefits that could occur with the convergence of system dynamics modeling and research into human disease, and encourage any reaction and discussion now. And as a follow-up, you have my contact information there. For those, I hope that there is a new wave of interest and work in this very promising and exciting arena. Thank you very much. Seth, over to you.
Thank you, Ken. That was fantastic. I really enjoyed that. So in answer to as we look at your closing question there, have you given thought as we wait for questions to roll in? And I'll take a moment of order here, please. To the people who are watching live, please do write your questions in the comments on YouTube, and we'll see if we can get those answered here live on the air. So Ken, how have you given some thought to how we do push this forward? What do we need? What's the call to action?
Well, one step is this platform that you have graciously provided to put that put that idea and question out there, welcoming all ideas. One of the things that I'm with several others in the System Dynamics Society working on now is a platform through which we can encourage the development of human disease modeling within the ranks of the society members. The current thought is to engage in some form of, as a start, a cancer modeling challenge, a call to arms, if you will, with awards and prizes and hopefully some very prestigious scientific support. But we're in the very early stages of this. This is a really in its embryonic form. And I think the important thing right now is to get the discussion going and hopefully to pique the interest and enthusiasm of those who are in a position either on the side of the current medical science field or in the modeling world, or perhaps those who have a foot in both worlds, to reach across and help bring about some early examples of this very valuable modeling.
Okay, excellent, excellent. So we do have one question that's come in. So I wanted to thank you for the inspiring presentation. What is your recommendation regarding who should be the ideal client for these models? Pharma manager, clinical researcher? What are your thoughts on the intended audience?
Well, I think given my background, my inclination always drifts quickly to ensuring that there is a commercial paying client that's willing to pursue the work, and so I think pharmaceuticals represent a tremendous opportunity, market opportunity. That's not to say that that research funding through a number of different agencies wouldn't also be possible, or a combination of both. But I think there's no shortage of client possibilities if only we can get the message out to them. This is something that's outside their comfort zone by and large, just as biological modeling is somewhat outside the traditional comfort zone of system dynamicists.
Mm-hmm. No, that makes sense. That makes sense. So a follow-up to that. So with your experience working with legal and policymakers, how would you suggest we introduce this approach to those groups of decision makers?
Well, I think that again, what we're doing here hopefully is a start. It's important to be able to get to some visionaries in the realm of take pharmaceuticals as an example, and you have to talk with a lot of people, expose a lot of people, is my experience, in order to find the one or two or three who gets it and can operate as an internal champion within their organization. And this is part of the outreach that I think a lot of us hopefully will be engaged in now.
Exactly. I'm sure we have in our networks members of these communities in one form or another, or a few degrees away from those individuals who can be those champions. And I think that's an important call to action for this audience that we have right now. If there aren't some of those decision makers who've tuned in today or who are watching post fact, can you share with us the approximate effort of what it took to put these pilot models together in terms of FTE hours or dollars, or some metric to set scale in this effort?
Sure. Let's see. I haven't stopped to add it up recently, but everything that I described today, and obviously all the things that ancillary efforts that went along with it, was done with about a dozen person years of effort. Okay. on that order. So all four of the modeling efforts I described today accumulated to about that much effort.
In total, not individually.
in total.
Got it. Got it. Wow. It's a pretty good return on investment, I would say, in terms of what you've been able to foresee in other forms of research that you brought out, in terms of how those insights have come about and become more accepted within the community years later, after you've foretold them with the modeling approach.
Well, it's encouraging, I think, not so much in simply proving accurate what we had foreseen, but I think it's a strong indicator of the power that good system dynamics modeling can bring to this arena of human disease research.
Next question for you. How did you overcome, did you encounter, first, I shouldn't presume the truth of this, but did you overcome, encounter prejudices and establish this is the way we do things? And if so, how did you overcome them from within the physicians and others within the scientific community, based on these analytic methods that you're proposing?
I'm smiling because we encountered almost nothing but prejudice. And I can't speak definitively to how all of them were overcome, because all of them were not. We had, interestingly, we had many who, the vast majority, who said that what we did could not be done. In fact, there were a few who, surprising to me, told us that we should not even attempt it. In talking with one, I'm reminded that in talking with the dean of one medical school, I asked him how they approach the teachings of how the immune system works, and his response approximately was, "Well, we're fortunate if we can teach all of the different components of the immune system. There really is no mechanism or means by which we can teach how the system operates as a whole." And of course, that's probably true. I think that whole arena is yet another possibility, the arena of medical education through the use of simulators, such as the kinds I just described, would be an enormously valuable tool for experimentation in 21st century medicine, medical training.
Absolutely. No, that makes sense. And carrying on the training element, the field of systems dynamics has benefited tremendously from the work on archetypes. How would do you think there would be similar benefit to expanding that collection to include cell models as well?
Cell models, certainly. There's so many different levels on which the system modeling can occur. Intracellular models, multicellular models, whole system models of the sort that I've described today would all be useful, and one can imagine and hope for, I think, ultimately a hierarchical system of models that, just as the Human Genome Project produced such extraordinary advances, that a systems model of the workings of the systems of the human body could develop over the course of some number of years to encompass many levels of operation within the body for I think what would be an enormous contribution to medical science.
No, I like where you're going there in terms of making that information, those dynamics, that understanding available in ideally in the public domain for those to, for a collaborative, very much like the Human Genome Project view of these dynamic systems.
Exactly.
All right, carrying on, the questions are flowing in. So next question. So your approach to identifying those levers, was that optimization driven, machine learning, Monte Carlo? How did you come to identify those levers in the particular combination?
Yeah, all of those. And in addition to, and so there were thousands. You'd turn on the model Friday afternoon and come back Monday morning to examine the results of thousands of simulations that had been run. We had to have some organized means of examining those results as well, and that was it was useful to have that. But all of those. But in addition to the mechanistic work, some good dynamic insight goes a long way as well, just from the minds of the modelers. And so some of the experiments were guided by our own thoughts about what ought to be interesting levers to try. But by numerical count, the vast majority were automated with all of those approaches.
Okay, so kind of intuitive, seeding the simulation to look in a particular direction, and then exploring the periphery around that, as opposed to examining the entirety of the parameter space.
Yes.
Okay. Let's see. Just reading the questions here, just a moment. So the question, I'll read it verbatim here. About your consulting experience, it looked like you showed, it looked like to show behavior modes of the model early in the modeling process. Did you calibrate the models, or just show rough order of magnitude?
Well, the answer to that is different for the different models, which we put together for different purposes. In the case of the immune system model that we built to examine the dynamics and treatments in HIV, we went through a large amount of data development, validation processes, which I hinted at in the slides, and showing the replicating documented behavior and results in clinical trials, and we kept a large sort of encyclopedia, if you will, of the with linked data sources to every parameter in the model that we could possibly do so. Mm-hmm. So in that case, there was a very, because we were looking for guidance on the clinical trial, there was a great deal of numerical validation on that case, as there could be with any of the models. The other three, for type 1 diabetes, CTE, and the tumor model, were behavior mode models that corresponded in magnitude, direction, mode of behavior to documented literature, basically to explore what kinds of possibilities there would be in examining each of those diseases.
Okay. Well, makes sense. Makes sense. Well, I think that's about all that we have time for. The questions have stopped. And I want to take a moment again. Thank you, Ken. Really do appreciate it. If people wanted to contact you, what is the best way to reach out?
Through email is the best bet, and that's kencoopersd@gmail.com.
Very good.
It is on the slide that is, I assume it will be available for people to see.
Yes, yeah, we can see it. We can see it on the screen now. So that's very timely and and reinforcing. So that was very good. So to the audience, if you joined late or want to review, today's episode will be available or is available on YouTube presently, and here on the Business SIG's YouTube channel. I want to thank Rafat Zaini for helping us manage the chat, and for helping to arrange guests for these series of webinars. If you're interested, to that end, if you're interested in sharing your work or know of someone who may want to present more, please do let us know, and we'll reach out and discuss that opportunity with you directly. Stay tuned to the Google group. We will announce our next webinar soon. Thank you for joining us, and please, as always, share, comment, like, subscribe, share this with your audience, with members of your professional networks, and because we need to get this information in front of the right people, and to build those relationships and put this to use. So with that, we will see you next month, and thank you for joining.